Good evening, everyone, and a very warm welcome to Dorset Humanists. Just to let you know, we are hoping to return to Moordown Community Centre on Saturday, the 10th of July. It's not absolutely certain yet, but that's what we're planning for. And I will, of course, let you know the details of that in due course. We should have a speaker, and we're hoping as well to put on a cream tea to entice you out of your hibernation. So 10th of July, hopefully, all being well, we'll go back to Moordown. We've just, Lynn was actually just mentioning a few minutes ago that we've just heard from the Greenhouse Hotel that their charge for an evening booking has increased from a very affordable £50 pre-COVID to I think something like £250 post-COVID. I think they're trying to recoup their losses. So that's going to be rather out of our price bracket for the foreseeable future. So we're going to have to find a new venue for our Wednesday evening meetings. And Lynn was saying earlier, if you know of anywhere suitable, somewhere that's similar to the greenhouse in terms of size and accessibility and affordability, and somewhere that is within easy reach of people in the centre of either centre of Bournemouth or Poole, or on a good bus route, something like that. So do let us know if you've got some ideas. We've got some ideas that we'll pursue, but let us know what you think. Okay, so we, as you probably know, now we are recording this event. So if you don't want to be seen on the recording, especially if you're still eating your dinner or your pudding, then do please turn off your video for the duration. Our speaker this evening is Greg Atkins. Greg is a member of Dorset Humanists, and he was a professor of virology at Trinity College, Dublin, before he retired to Somerset. And Greg was saying to us a few minutes ago that when he was working in virology, no one was interested in the subject, but for some reason it's become a topic of great interest. So this is Greg's second talk for us on coronavirus. He gave us a great talk last year as we were sort of going into lockdown. So we're going to see what he's going to say to us this evening, a year on from when it all started. So Greg, thank you very much for joining us this evening, and I will just unmute you, and I will then hand over to you, and you can start showing your slides. So let me make sure that we can hear you, if I can find you on the screen.
I've unmuted.
Oh, you are unmuted. Okay, Greg.
Yeah,
well, if you...
All right, so I'll try sharing then.
Yeah, great.
Hold on now. Screen broadcast, yes. Okay, you can still hear me, can you?
I can hear you, but yes, that's looking good.
Okay, now, hold on, sharing the screen. Doesn't seem to, oh dear, what's happening now?
Don't worry, we'll...
Okay, can you see my screen?
Yeah, we can.
Oh, right, okay, that's it then. Let's just go to PowerPoint. Okay, so that should be coming up now.
That's coming up.
Okay, can you see the slides now?
That's it, yeah, great.
Okay, so from start. All right, so I can't see any of you. I can only see my my slides at the moment, but I'll assume you can hear me. Am I right?
Yeah, that's okay. Fine, Greg. Yes, fine. Just go ahead.
All right. Well, as David was saying, I gave you a talk on this a year ago, and I was actually, before we started, I was talking to Aaron, who had this idea about talking about coronavirus, and he was very astute because he picked up this virus when it first occurred in China, and he said we should have a talk about this virus, and he asked me if I wanted to give a talk about it in March, and this was in November, I think, November or December, and I said, well, it'll be old news by then, there's no point in giving a talk, and David and Aaron were quite insistent I should give it, so I gave it, and lo and behold, look what's happened. As David was saying, I was a virologist for many years, and whenever I mentioned what I did for a living, people kind of adopted a bored expression and changed the subject. And I remember actually first meeting David when I first joined Dorset Humanists. He said, "Oh well," I mentioned what I did for a living, and he said, "Well, can you give a talk?" And I said, "Yes, I don't mind giving a talk." And he said, "What's your subject?" I said, "Virology." And I said, and he sort of, I could see by his expression that he was not very interested. So I said, I did give one lecture on evolutionary ethics. And he said, "Oh, we'll have the evolutionary ethics." So just shows you how things have changed. People now are quite interested in this subject, as you can imagine. Now, I retired officially in 2009. But I carried on working, even though I was officially retired up until 2014. And I came to England then. I was living in Dublin up or near Dublin up till then. I came back to England, and I retired completely then. So that was what, 7 years ago now. So I'm a bit rusty. So what I'm relying on really is my elder son Tim, who works at Porton, and he's been organising part of the response, of the emergency response at Porton, including the coronavirus response. You might have seen him played by an actor, actually, in the documentary on the Salisbury poisonings. He was played by a Shakespearean actor, which is quite a turn up for the books. But anyway, I'm going to talk about what's happened since I gave the last talk. So what I'm going to do is to give a little bit of a recap, just to refresh you, but I'm not going to go over what I said in the last talk. I'm just going to try and set the stage for what I'm going to talk about now. And what I'm going to do is to talk about developments since I gave the last talk. So there's been three main areas of development with this virus. First of all, diagnostic tests, which I'm going to say a little bit about. And then I'm going to talk about the development of vaccines, which has been quite a success. And then about these new variants and try and set the stage there. So first of all, a little bit of a revision as to what coronavirus is. As I mentioned last time, there are two big groups of viruses: DNA viruses that have DNA as their genetic material, just like we do, and RNA viruses, which have RNA, and they're unique in that they're the only living organisms, if that's what you call them. Some people debate whether they're really alive, but RNA viruses are unique in that they have RNA as their main genetic material. We have it as a sort of secondary function to DNA. So coronavirus is an RNA virus, and it is actually called, the virus is called SARS-CoV-2, which is the designation by the International Committee for the nomenclature of viruses, and we all know it's usually called COVID-19, but that's actually the name of the disease, not the virus. And this was designated by the World Health Organization as the name of the disease that coronavirus causes. Now, it's related to two other viruses which caused outbreaks in the early on in the 2000s. They were the SARS virus and the MERS virus. Both of these were very limited outbreaks, and just a few hundred really of casualties. And so SARS-CoV-2 is related to these, the virus causing COVID-19. It's also related to viruses that cause about 20% of common colds. Coronaviruses are a big family of viruses infecting a lot of animals as well as humans, and they cause about 20% of common colds. And in the last talk I gave, somebody asked me a question, I remember, as to whether you could be immune to this coronavirus if you've had a cold caused by coronavirus. I said no at that time, because that was what the general information about this was. But it turns out that there may be some cross-reaction now, and that some people who are at least partially immune to coronavirus without previously being infected might be immune because of being infected by a common cold virus. So that's now become a possibility, although it's not a common occurrence, I don't think. Now, the other thing that's changed that seemed to be certain when I gave my last talk was its origin. And you may have read something in the news today about this. It was assumed when I gave my first talk that the origin of this virus was Wuhan market, where they were selling all kinds of bush meat and animals for food. That is coming increasingly under doubt, I'm afraid. Now it seems that two possibilities are being investigated. And it may not be Wuhan market at all that was the origin of this virus. What I think can be ruled out is a theory that this was a virus engineered by the Chinese. There are only certain ways of making a new virus. You can do it in the lab, it's been done, but it always leaves footprints. If you make a virus from bits of other viruses, which is the way you have to do it, it always leaves footprints, and this virus doesn't have such footprints. So it wasn't probably, it's very unlikely to have been engineered by the Chinese as a germ warfare agent or something like that. That probably didn't happen. But there is this possibility that it could have escaped from a Chinese lab, because the Wuhan Institute of Virology was actually carrying out investigations into coronaviruses that infect bats. And also, you may have read today in the newspaper that it's only just become apparent that in 2019, several workers at this Institute of Virology were actually hospitalized with a disease which seemed to be very similar to COVID-19. So there is, unfortunately, that possibility. Now, the other possibility, which I think probably is more likely at the moment on present evidence, is that it probably didn't come from Wuhan market, because some of the early cases that the Chinese described now appears had connection with Wuhan market and couldn't be traced back to anybody who had connections with Wuhan market. So it appears that another possibility is that this virus got into the human population sometime quite a long way before it was actually detected, and that it gradually evolved to infect humans more and more efficiently, and finally ended up by spreading the way it did. And as you'll see later on in the talk, this process of efficiently is probably still ongoing. So that's another possibility. How did it actually arise? Well, it's pretty clear from looking at the sequence of this virus that it's a hybrid virus. It's made of bits of coronavirus sequences from other origins. And the primary source of the virus appears to be from a fruit bat, but it's not entirely of that sequence. So it probably came from a bat, probably a fruit bat, but it does have sequences that suggest that it's been through another animal as well. And one possibility, in fact, is an animal called a pangolin, which is a kind of Asian anteater, which is traded a lot as bushmeat. But it could have been another animal, we don't know. So it's not very satisfactory, but there are those different possibilities for the origin of the virus, and it may not have come straight from Wuhan market. Okay, so let's just revise then what coronavirus actually is. This is an electron micrograph of coronavirus. You'll remember from the last talk that you can't see viruses in a normal microscope, they're too small. The size of coronavirus is 100 nanometers, and to put that in units that you can understand, that is 1/10,000th of a millimeter. So it's incredibly small, and you can't see it with a light microscope. So you have to use a different kind of microscope called an electron microscope. The reason you can't see it in light microscopes is that the wavelength of visible light is too long to be able to see such a small particle. So electrons have a much shorter wavelength, and so that's what's used to visualize viruses. And this is an electron micrograph of coronavirus. You remember from the last talk that electron microscope works by like an old cathode ray television. It puts a beam of electrons through the specimen onto a fluorescent screen originally, but now it's done using computer technology. But what you always get out is a grayscale image like this. You'll see images, colored images of coronaviruses, but that's just been put in artificially to make it look pretty. This is how the electron micrograph turns out. You can see from this that they are bags of membrane, if you like, with the RNA in the middle of the bags, and protruding from the outside of the virus, and this is very important, are these spikes. And the function of these spikes is to latch on to susceptible cells. The reason it's called a coronavirus is because of this ring of spikes. It looks like a crown or a corona. So that's where the name came from. So this is just a diagram of what the virus looks like. As you can see, it shows the envelope with the protruding spikes, and the one called the spike glycoprotein is the one which is of primary importance, and I'm going to talk about a bit more. This is the structure which latches onto susceptible cells. Now, how does this happen? Well, this is a summary of the way the virus multiplies. The virus attaches to a protein on the cell surface which has a completely different function. It's a protein called ACE2, and this is the receptor for a human hormone called angiotensin. And the normal function of this hormone is to regulate blood pressure and other aspects of the circulatory system. So it normally has a completely different function, but the virus hijacks this protein, if you like, and attaches to it. Now, the ACE2 receptor, as it's called, is found on many different kinds of cells throughout the body, but it's particularly prevalent in lung tissue. And of course, the coronavirus is particularly is really a respiratory virus, although it does infect a lot of other organs in the body as well. Okay, so the virus gets into the cell, it replicates its RNA, and it makes its proteins. these proteins get wrapped around the RNA, and then it's released from the cell, and it picks up a bit of the membrane material of the cell as it leaves, and this forms the envelope of the virus, which surrounds the RNA, and the spikes have been inserted into this envelope, which is derived from the cell membrane. So that's, in a nutshell, how the virus multiplies. And of course, this process is very disruptive to the cell. And the fact that it can kill cells or greatly modify their function when it does this is one of the ways coronavirus causes disease. Many other ways as well. Okay, how has this affected us in the UK? Well, it's normally said there's been 2 waves of coronavirus, but as you can see from this diagram, most people would say it was 3 actually. But the accepted knowledge is that there's been 2, but I can see 3 peaks there. I don't know about you, but there we are. Okay, so how has it affected this country? Well, I did a bit of Googling just before this talk began this afternoon because I was a bit bored, just to put it into context. Now, we all know there's been about 127,000 deaths in this country due to coronavirus. That is the 5th highest in the world. And there's been a lot of comparisons made with India at the moment. Our death rate due to this virus is about just very nearly 200 per 100,000 population. In India, it's around 20 at the moment. So we're much higher. And in fact, it's quite a disaster, because you're talking about the deaths of 2 per 1,000 people of the UK population from coronavirus. And as I say, I did a bit of Googling too, and just to amuse myself this afternoon. And just to put it into context, this is nearly twice the number of people that were killed by German bombing in the Second World War, and 10 times the number of people that were killed in the bloodiest day of military history. That is the first day of the Battle of the Somme. So it's really an unmitigated disaster. Okay, so how is this virus transmitted? Again, a bit of rethinking here since the last talk I gave, because you have seen all this business about, what do they call it, hands, face, and I can't remember what the other bit was. But the main mechanism of transmission is by aerosol. That is, when you're breathing out small droplets all the time. When you cough and sneeze, these greatly increase, and these can infect other people. Now, there's been a lot of discussion, and you can see this happening here on this photograph here. There's been a lot of discussion about washing hands, but I've looked at the literature. I can't see any description of this virus being transmitted by touch, and the technical term here is fomites. It's just not been described, and it's not been proved to be possible. My son in Portland is trying to see whether this really is the case at the moment. But we've had all this emphasis on washing hands, but I suppose it's a good thing to do just on the off chance. But I wonder whether it really is involved in any way in transmitting coronavirus. What is involved for sure is aerosol transmission. And of course, being in an enclosed atmosphere, an enclosed space indoors, greatly increases the chances of that happening. And of course, if you wear a mask, it cuts down on transmission by this means, cuts down mainly you transmitting the virus to other people. So that for sure, but I don't know about touch. the jury's still out on that. Now, just to finish off with this sort of revision of what I said before, of course, there's immunity to the virus. And we hear a lot about antibodies. These are specific proteins made in the blood which neutralize the virus and also act in other ways. But they're only one aspect of immunity against the virus. And they may even not be the main mechanism of immunity, because there's another mechanism of immunity, which is cell-mediated immunity. These are specialist T cells in the bloodstream which kill virus-infected cells. And this, again, is specific. And this is a very important mechanism of immunity. And the third arm of the immune system, if you like, is the inflammatory response. And this is non-specific. It's similar to what happens with many different kinds of infections. And the inflammatory response is a complex series of events involving a whole lot of different mechanisms. But the main point about the inflammatory response is that it can go wrong very easily, particularly in older people. And many of the people, probably the majority of people that die from coronavirus, die because of this inflammatory response going wrong. So there are those three kinds of immunity that are developed against virus infection. Now, okay, I want now to start on considering aspects of developments since I gave the last talk. First of all, the diagnostic tests that are used for coronavirus. Now, we all know about the gold standard test, which is called the PCR test, and that stands for Polymerase Chain Reaction Test. This is it in a nutshell. You take a swab from the nose and usually from the throat as well, put it into a buffer, a liquid, which disrupts the virus and releases all its components. And then you use a machine. Its popular name is a PCR machine, but its actually technical name is a thermocycler, and it carries out the reaction to detect the virus. I'll show you what this is in a minute. The main point about this test is that it can only be performed in a lab because it needs this very specialist piece of equipment, and also the reaction in itself takes several hours. So it's usually several hours before you can get a result of this test. But it is the gold standard. Its main drawback is that it's of medium sensitivity, and it does produce some false negatives, particularly people in the very early stages of infection. So it's a highly accurate test, but a very low proportion of false negatives. Now, the test that's been introduced more recently, this is the way the RT-PCR test works. It's usually just called a PCR test, but the RT stands for reverse transcription to describe the first step in the reaction. It detects the RNA of the virus, and in order to detect this RNA, you must first convert the RNA to DNA, and you see the first step in the reaction there. This is carried out by an enzyme called reverse transcriptase. It's called reverse transcription. That's where the RT bit comes in. So you make DNA from this RNA. You then use small pieces of DNA called primers, which bind specifically to viral DNA, the viral DNA that's been produced. You use an enzyme called a DNA polymerase, and what this does is to amplify the sequences between these primers. And this has to be carried out at a specific temperature, hence the piece of equipment. And what this what this does is to incorporate the dye into the DNA that's produced, and you can measure the quantity of this dye that's been incorporated. And from that, you can tell whether the sample is positive or negative if any dye has been incorporated. But the other advantage of this test is that it's quantitative. You can tell how much RNA is there. But it has another very important advantage as well, in that you can sequence the DNA product. So you can tell what kind of virus you've detected, whether it's specifically what kind of variant it is. In other words, whether it has a sequence that's a bit different from previous virus sequences. So that's the big advantage of using this test. But it requires specialist equipment, and it takes a relatively long time, although it is the gold standard for detecting coronavirus. The one that's been produced more recently is this lateral flow test. This works on a completely different principle. The way this works is to detect virus protein rather than nucleic acid, rather than RNA. So you put the sample into a buffer which disrupts the virus, the same as before. But what you're looking for now are the virus proteins that are released, not the viral RNA. So you use a slide like this, and you put the sample on one end of the slide, and the proteins diffuse and they react with synthetic antibodies which are present in the slide. And these antibodies have a dye attached to them, and if you get a red band where it says T there, that stands for test, that means that the virus is present. There's a control as well, so you've got to have this band called C to show that the test is actually working. And so this is a very easy test to do, and it only takes a few minutes, about 20 minutes. So this is a test which is much easier to do than the PCR test, and it can also be done, it doesn't need a specialist laboratory. It can be done, for example, in schools or even in homes. The problem with this test is that it does produce false positives, and the government will have us believe that the number of false positives is only 1 per 1,000. But this seems to be disputed somewhat. And it seems to me that what's happening here is it depends on who's actually doing the test. If you've got the test being done in a laboratory by a skilled technician, probably you're only going to get 1 in 1,000 false positives. But if this is done by somebody who's had no experience of the test before, is done outside of the laboratory, it might give you many more false positives, and that seems to be what's happening. Now, the sensible thing to do, of course, if you get a positive result from this, is to go and have a PCR test done. But unfortunately, the government wasn't recommending that. They were recommending that people who get positive tests using this test should go into isolation, which seems to be a bit peculiar. But anyway, that's the lateral flow test. So there are these two tests which are being used at the moment. I understand you can get a free lateral flow test now from chemists, and you don't have to pay for it. So, so this is quite useful, but it does have its disadvantages. Okay, so those are the two tests that are developed. What about ways of treating the virus? Well, let me say straight away here that antiviral drugs in particular have very limited applicability compared to, say, antibiotics against bacteria, which at least they were of very widespread applicability. It's getting more difficult now, but antiviral drugs, there are only quite a small number of them. really this is because of the way the virus multiplies in intimate association with the cell. So it's very difficult to get a drug that acts only on the virus. But there have been some developed, and there have been some which are being used for coronavirus. Now, the one that's one class of drugs that are being developed are drugs which dampen down the inflammatory response, because, as I said before, this is the way that many people die from coronavirus infection, because of the inflammatory response going wrong. So drugs have been tested, many drugs have been tested, but only one really has turned out to be of widespread use, and that is dexamethasone, which was used originally to treat arthritis, but it turns out that it is quite useful in treating the inflammatory response due to coronavirus. And really it's useful in preventing people that are already sick from coronavirus, from progressing to intensive care or indeed dying from it. So that's the usefulness of dexamethasone. The other class of drug that's been developed are drugs which target the viral RNA polymerase. That's the enzyme that copies the viral RNA. And these are drugs called base analogues. They mimic the components of RNA, but they screw up the RNA when it's synthesized. They're not actually the components that are really meant to be used. So one drug that's been developed, which is of this type, is the drug called remdesivir, which is used extensively to treat coronavirus. And again, it's useful in the same way. It's usually used for people who are already sick with coronavirus. So really, it's only these two that are in widespread use at the moment, but there are others being developed. One class of drugs which isn't yet in widespread use, but which could turn out to be, are drugs called monoclonal antibodies. These are synthetic antibodies, and they are designed to neutralize the virus so it's no longer infectious, or to block the ACE2 receptor of the virus. So there are a number of these being tested at the moment in clinical trials. None of them yet have been developed into widespread use. One thing that's been tried here, and you think would be effective, would be serum from people who have recovered from a virus infection, and to use that to treat people. That is blood plasma. That's serum from people who've recovered from the virus, and which should contain antibodies. Now, there have been quite extensive clinical trials done using convalescent serum from people that have recovered from virus infection, but it turns out it has very little effect. And I think this underlines the fact that antibodies are not the main component of immunity to this virus, that other components of the immune system, particularly cell-mediated immunity, are just as important, if not more important, and they probably act in conjunction with antibodies. So it turns out that convalescent antibodies don't seem to be of any use. So that was one dead end. I could mention a few other dead ends, actually. We all remember the fuss about chloroquine, which was touted by Trump as being useful for coronavirus. Turned out it was based on a very small clinical trial which couldn't be reproduced, so that turns out not to be of any use now. In terms of experimental drugs which might be useful. I mentioned monoclonal antibodies. Interferon beta seems to be a good candidate. This is an immune modifier. It's a protein called a cytokine, and it's actually been used to treat multiple sclerosis. But it's both antiviral and anti-inflammatory. So that has potential. There are clinical trials underway with that at the moment. And another one that I could mention is this drug called Ivormectin. I didn't see Chris Street here, but I know he's been involved with this. And it's something that's been politicized, just like chloroquine was. And people in the States in particular have been poisoning themselves with ivermectin because, again, there was a very small clinical trial which seemed to suggest that it did have some activity against the virus. It turns out that 2 other small clinical trials now have shown no real effect. Studies on the virus infecting cultured cells have shown that ivermectin does inhibit the virus, possibly by blocking the receptor. But real clinical trials really have not shown anything much. Ivermectin is actually used as just like chloroquine, which is used against malaria parasites. Ivermectin is an anti-parasite drug, particularly used used in veterinary use, like, for example, parasites of horses and dogs and cats. So people have been trying to treat themselves with this drug meant for horses and have been poisoning themselves. But the WHO and 2 drug companies have declined to conduct large-scale clinical trials with this because they say the preliminary evidence isn't there. So that's the story of ivermectin. Just like chloroquine, it seems to have been politicized. Okay, how are we doing for time? 3/4 of a minute. Okay, so let's now go on to vaccines. And this has been quite a success story, particularly in this country. I should have said, having given that very somber assessment of the way this country dealt with the virus in the first place, we've done very well as regards vaccines, and our vaccine rollout has been the 3rd best in the world, I think, after Israel and I think it was Dubai was the. So it's been very good so far, although other countries are now catching up. Vaccines used in the past have been of 2 types. Live attenuated, like the MMR vaccine. This is live virus, but which has been weakened, and like the common polio vaccine. Then there's inactivated, like the Salk polio vaccine. These are chemically inactivated viruses. The flu virus vaccine that older people get is of this type. But there's a 3rd type that's been recently developed, and these are vaccines which are produced synthetically by genetic engineering, by genetic manipulation of the virus. And this is a very new thing. The first time it was tried on any large scale was during the Ebola virus outbreak in the Congo, not the original one, but more recent outbreak in the Congo, and it was very successful. It turns out that the vast majority of coronavirus vaccines which have been developed now are of this type. There is one exception, which I'll tell you about. Now, my son, Tim, who's been looking at this at Porton, tells me that at the last count, there were 84 different vaccines being developed in the world. So this has been quite an effort. So what are they? Well, at the moment, you all probably know, and you've all probably had these already. I have had the Astrozenica, and I expect other people have had these vaccines as well. These are the three that have been approved in the UK. Only three at the moment: the Oxford AstraZeneca vaccine, the Moderna vaccine, and the Pfizer vaccine. The Moderna and the Pfizer are very similar vaccines. The AstraZeneca is completely different. They all require 2 doses to bring to give maximum protection. Now, what are they? Well, this table just shows you how effective they are. It's very this figure that's given for how effective the vaccines are is a bit misleading, because it's saying how effective they are at stopping any kind of infection, and this includes mild infections or mild asymptomatic infections. Turns out that in the clinical trials anyway, there were no hospitalizations and no deaths of people who received these vaccines. So they're in fact very effective, all three of them. The big difference comes from practicalities in these vaccines. The Oxford AstraZeneca vaccine can be stored in an ordinary fridge, and from this point of view is a very practical vaccine, in that it could be used, for example, throughout the world in many different countries, particularly developing countries, without much trouble. The other 2 vaccines, these are RNA vaccines, which I'll come on to in a minute, exactly what they are. They need special storage conditions. The Moderna one, which is, I think has been approved in the UK, but not used very much yet, that could be put into an ordinary freezer. But the one that's RNA vaccine, which has been extensively used here, requires a special low temperature storage freezer at -70 degrees, and really these ultra low temperature storage cabinets, which cost around £10,000 each, are not usually found in places like GP surgeries and places like that. They're usually only found in specialist laboratories. When I was working, we had 3 of them actually, but they are expensive and they're not found everywhere. So that's a disadvantage of that vaccine. Can be stored for short periods in an ordinary fridge, but you're talking about very short periods, maximum of a couple of weeks at the very most. The other big difference between them is their cost. The Oxford AstraZeneca vaccine only costs around £3 a dose, whereas the Moderna and Pfizer vaccines are very much more expensive. As you all know, the Oxford AstraZeneca one is the one that's been used for the majority of people here. What do they actually consist of? Well, first of all, the Oxford AstraZeneca vaccine. What this is another virus, and what they've done is to take a chimpanzee virus similar to, it's called an adenovirus. It's a DNA virus, and it's similar to viruses which cause around 10% of common colds. This is a DNA virus, which you can see its structure on the left as protruding spikes. What they've done is to copy the spike protein of coronavirus and to insert this into the genome of this chimpanzee virus. And the chimpanzee virus has actually been further debilitated by deleting bits of it, but it's still infectious. So this virus by itself won't cause any disease, but what it does is it causes the cells it infects to make large amounts of the coronavirus spike protein. So it's a genetically engineered virus. It's a coronavirus gene inserted into a completely different virus. And the gene that's inserted is the gene that forms the coronavirus spike. To understand how the other 2 vaccines work, the RNA vaccines, you've got to understand a little bit more about the way coronaviruses multiply. Coronaviruses are the largest RNA viruses known, and they use a very peculiar mechanism for producing their RNA called a nested set, like a set of Russian dolls. Each RNA includes the sequence of the previous one. That's why it's called a nested set. You will see on this diagram one that's labeled S. That is the RNA that codes for the spike protein. So what the Moderna and Pfizer vaccines have done is to take a synthetic version of this RNA that codes for the spike protein. So it's mimicking a natural RNA of the virus. So how does it work? Well, what they do is wrap it in a kind of fatty material that makes it like a virus. So when you inject this into somebody, this wrapped RNA actually acts like a virus, and the RNA gets into cells. And what it does is it acts like a normal RNA from the virus. It starts to make spike protein, but it only makes spike protein. It doesn't make any other bits of the virus. These spikes are the synthetic spikes which are made inside the cell, are then inserted into the cell membrane, and they stimulate immunity quite efficiently. So this is the way that works. It works in a different way, obviously, to the Astrozeneca vaccine. So how many doses has the UK ordered? Well, it's a complete overkill here. 100 million doses of the AstraZeneca, 40 million of the Pfizer, and 17 of the Moderna. So we have far more vaccines, even at the moment, of the 3 approved vaccines than we actually need. There are these various other vaccines which have been ordered. I'll discuss what they are in a minute. Just to pick out one, the Janssen one. This is marketed by Johnson & Johnson. It's one of the main ones used in the United States, but hasn't been approved or used here yet. It's very similar to the AstraZeneca vaccine, but they say it only requires 1 dose. It's used quite extensively in the United States. Okay, so what do these vaccines consist of? I mentioned the AstraZeneca, I mentioned the Moderna, I mentioned the Pfizer bio vaccines. These are the 3 that are used here. There are also 4 that are used extensively in other parts of the world, not used here at the moment. The first one on this list is the Russian one, the Sputnik 5 vaccine. This is very similar to the AstraZeneca in that it uses an adenovirus vector. So that's similar to AstraZeneca. Now, the Sinovac vaccine, used quite extensively in the world, obviously used in China as well. This is the Chinese vaccine. This is the only one that uses the old technology. This uses chemically inactivated virus. So this is whole virus grown in cell culture, which is then chemically inactivated. Very similar to the technology that's been used extensively in the past for other vaccines, but it's not used here. This technology is being developed here by a company called Valneva, which, but it hasn't been approved yet. And then another one that's been ordered in the United Kingdom is the Novavax vaccine. This is completely different to all the other vaccines. What this is it's purified spike protein. Normally, purified proteins don't stimulate a very good immune response if they're just injected by themselves. But this is injected with a chemical which is designed to stimulate an immune response. It's called an adjuvant. And it's a substance called saponin, which is a complex carbohydrate. So if you inject the spike protein, the synthetic spike protein, with this adjuvant, you do get a good immune response, but this hasn't been approved here yet. And then finally, I mentioned the Janssen vaccine, similar to the Astrozeneca, which is used in the States, marketed by Johnson & Johnson. So, oh, hold on. How do these compare? Well, the adenovirus ones are the ones that could be stored in an ordinary fridge. The other ones, the RNA vaccines, require low temperature storage. So that's really the difference between them. Finally, how are we doing for time? Oh dear, I've got to hurry up a bit. Okay, I'll just quickly consider the final thing I want to consider, which is coronavirus variants, which have caused problems recently. Now, when the virus multiplies, it mutates at quite a high frequency. The reason for this is because of the way the RNA is copied, the RNA polymerase. RNA polymerase makes mistakes very regularly. And these mistakes, of course, are mutations. Most of these, and these are the raw material of any kind of evolution. But the enzyme that copies the RNA in coronavirus is error prone. It makes mistakes, and it does this at fairly high frequency. The reason why it makes so many mistakes is because it hasn't got a proofreading function. Once the mistakes are made, they can't be corrected. The enzyme that copies our DNA can correct mistakes, and that means that far fewer of them actually get through. But with coronavirus, it makes lots of mistakes. This doesn't normally matter to the virus because it makes so much RNA that there's far too much that is needed to reproduce the virus. So a few mistakes won't matter. But they do have significance because sometimes, very occasionally, they give the virus an advantage. And you can see here a diagram. This was work that was done at the Sanger Institute in Cambridge, and where lots of different isolates of coronavirus have been sequenced, and each of these dots represents a different variant. You can see there's a huge number, several thousand have now been detected, and there's probably very many more than that as well. But you can see the green dots represent mutations which either have no effect at all or which actually are detrimental to the virus. But you can see that there are a few mutations which actually confer an advantage on the virus. And there are three that are marked here. The South African variant, the UK variant, which we have predominant at the moment, and the Brazil variant. We could add to this now the Indian variant, which I'll talk about in just a second. Now, you've got to understand as well the selection pressure on the virus. During the lockdown, the spread of the virus is inhibited deliberately to try and get fewer people infected. Now, the virus has responded to this because what has happened is you've selected out variants which are able to spread more rapidly. And this is what's actually happened with virtually all the variants that have been produced so far, which have an advantage. These are all variants which spread more rapidly. The lockdown has actually been quite effective because the amount of influenza, for example, that's prevalent now is almost zero. So it's been very effective. So there's a tremendous pressure on the virus to be able to spread more efficiently. And that's what's happened. But we have a different situation now. We have a situation where some people are immunized and some people are not immunized, and are susceptible. So the virus can multiply, but it's under a selection pressure from people that are immune. It's under selection pressure from the immune response. If you remember what I said last year, influenza virus responds to this by evading the immune response, by producing variants which evade the immune response. That's why we have to have a different influenza vaccine every year. And the worst possible situation for selecting for such variants is what we've got now, a mixture of people that are immune and are not immune, not only in this country, but in other countries. So it hasn't happened yet to a large extent, but the selection pressure is there for what are called immune escape variants to occur. So what has happened? So these variants that give the virus a selective advantage are actually called variants of concern to distinguish them from other variants which are not of any concern. So ones that actually have occurred that can spread more easily. Some have occurred which might have higher lethality, but there's no selection pressure on the virus to produce a lethal form. That seems to occur just as a byproduct of something else. But the one that we really would scupper things to a big extent are if these variants occurred, like they do with influenza, variants that can escape the immune system. So these variants have actually been mapped, and we know where these mutations have occurred. They've mostly occurred in the spike protein again, and they enable the virus to attach better to susceptible cells. Oh, this is a complicated slide, but it shows you some of the variants of concern. There are a lot of them. I think the Indian variant, third from the bottom, is on that slide. Now, this variant does spread more easily. It's the one that has been replacing, at the moment, the current variant. It does escape the immune system to a certain extent, in that whereas the vaccines are about 80% efficient after the third shot for example, the UK variant. For the Indian variant, they're only about 30% efficient. So it has got, to some extent, a method of escaping the immune system, although the vaccines still are effective against this variant if you have 2 shots. But you can see how this could happen. So I want to end on this, and this shows you something else which is happening, which is quite concerning. This was a slide I got a short time ago when the Indian variant was first appearing in the UK. And from this diagram, you can see the very dark red areas are areas where it's prevalent at where it's present at high prevalence. These are actually areas where there's a large indigenous ethnic Indian community. But you can see they're scattered around the UK. They're not in one place. So this would suggest that this variant has actually been introduced into the UK on multiple occasions, not just one occasion. So this I think is going to be a big problem, importing variants like this. There seems to be no effective way of stopping it at the moment. Okay, well, I think I'll finish there, and on that kind of pessimistic note, I'll ask if you've got any questions. Okay, so I hope that summarised some of the new developments.
Thank you very much, Greg. There's plenty of material there for us to chew on. Let me pick up some of the questions that have been put in the chat, and then if people would like to ask a question, the easiest way to draw your attention to me or to get my attention is to go to participants and use the raise hand function. Some of you have already found that. If you wave at me, I might see you or I might not. Greg, let me just pick up 3 of the questions that were in the chat as you were talking. Sophie asked a question about the furin cleavage. I have no idea what that is, but do you know what furin cleavage is?
This is a technical way the virus produces its protein. I'm not quite sure what the question is about, but this is
Sophie, she said, does it indicate human interference?
No, I should have said that. One of the ideas as to how I presume this is what the question is about. I'm a bit unclear, but one of the ideas about the origin of this virus that was particularly touted by Trump and company was that this could have been produced in a laboratory. Now, my son has been, as you can imagine, has been quite seriously involved in looking at this. And when you attempt to make a virus for germ warfare purposes, which is, what Portal were originally about, in the lab, there are only certain ways of doing it. And when you do that, you have to use enzymes called restriction enzymes, and they have recognized particular sequences in the virus genetic sequence. So if you construct a new virus, you would leave footprints that could be detected. They've been looking quite extensively to see if those are there, and in fact they're not. So at the moment, you can say that there's no real evidence that this is a synthetic virus. I don't know what the question was about. Or was it about?
Sophie, does that answer your question? You can put your thumbs up or unmute yourself.
I'll unmute myself. Yeah, there are now a number of scientists, respectable virologists, who have changed their minds about this, because originally they wrote in The Lancet and a health science magazine saying that they denied the lab leakage possibility. It's a biochemical weapon. It's simply that a mistake was made because they weren't using level 4 biosecurity. They were using level 2 or 3. Yeah. And the lab had been ticked off several times for that already. And it's not about a biochemical, it's just a mistake that was made in doing the gain of function research, which had been done in the States as well and was being done in Wuhan. And as I understand it, the furin cleavage is a very clear sign of human interference because it doesn't occur in any natural bat coronaviruses or in the pangolins or in the dromedaries, which it had passed into before. And there are now respectable scientists coming out saying that this needs to be looked at again as a serious thesis. And it was linked to Trump and became political, but that's because he's an idiot and he didn't know. He was just poking a finger in the air. But now serious people are saying that this is a possibility.
Yes, I think I read today that the CDC in the States had dismissed that possibility, but yes, you may very well be right. I don't know the particular details of that particular idea, but the idea that this virus could have escaped from a lab has not been dismissed. That's still under investigation, and certainly at Portland they're taking that seriously, and I think they're taking it seriously at CDC in the States as well. The idea that this could have been a synthetically produced virus that was constructed by taking bits of different viruses and putting them together, I think that can be largely ruled out. Now, of course, there are other possibilities, the way this virus could have been manipulated. Yes, you're probably right, yes.
Sophie, can I just ask you also just to say something you also put in the chat about the UK being 17th in the world league tables rather than 5th, and you're saying that's something to do with the way we count?
Well, it's not. But if you look at the Worldometer COVID statistics, we're actually 17th. I mean, some of those are quite small countries, but some of them are significant countries as well. But also it's because we measure deaths by with COVID. So you could have had a car accident and then had COVID and then died of the car accident and still go down as a COVID death. So we have a very, very strict standard, which means we're pushed up the legal test in terms the league tables in terms of tests per million. And we don't know how other countries are measuring their death rate. It's probably not by the same rate. So we're self-flagellating, saying we've done really badly, but that is not actually clear at the moment until we find out how other countries have measured their COVID stats.
I don't know whether that's actually true. I thought that as well, because I had a conversation, an email conversation with actually David Nunn about this. What we do is we say that that somebody's got to have died within a certain length of time after a positive COVID test, and they could have gone for the test and be hit by a bus going out the going out the test centre afterwards, and that would be counted as a death from coronavirus. You're quite right, but it's completely different to the way, for example, that influenza mortality is measured. PHE measures influenza mortality as excess death. In other words, deaths over and above what you would normally expect. That's been the tradition. I've no idea why it's been altered now, but my son has actually showed me figures of coronavirus mortality using both methods of calculating it, and they're actually not much different, believe it or not. Now, other countries use different criteria, so it's difficult to see the wood from the trees here. For example, Belgium was classified as the worst in the world, but they use a different method. Their criterion for a coronavirus death is just that it's got to be recorded as such by a physician on the death certificate. That's all that's required. So it's a different method again. But as regards the way we do it compared to the way flu was done, there doesn't actually seem, although they sound quite different ways of measuring deaths, when you actually look at the statistics, there's not much difference.
Okay, thanks, Greg, and thanks, Sophie, for those questions. Let's go to Roger. I know you put something in the chat earlier about false negatives. I don't know if that's your question or you want to ask something else.
Well, I wanted to ask a second question. The one I put in the chat was, we were being told that the lateral flow test can provide false positives. Can they also provide false negatives?
I think that's much rarer. I think it does occur, but it's much more common to get a false positive from that test.
Fine, thanks very much. The question I wanted to ask, because I put my hand up, was we're told that people who have had both their jabs are less likely to be ill, but do we know what the statistics are on that? I mean,
yes. No, we don't, is the short answer, but this comes from the clinical trials where about 20,000 people were looked at. So it's not a vast number, but in the clinical trials, I believe there were no deaths and no hospitalizations. So that's where that comes from.
Okay, thank you very much. I'm afraid I'm going to have to leave you now, so I hope the rest of the meeting goes well. Thank you.
Thanks, Roger.
Bye-bye.
Let's go to Sally next. Sally.
Hi, yes, my question was with regard, you were saying that we were vastly oversubscribed with vaccine as a result of, we'd ordered far more than we actually could use at the moment. And I was wondering if there was any chance of that vaccine finding its way to places where they don't have enough vaccine. I mean, even if we only gave it away, it would still be better than it not being used at all.
Well, you're getting on to ethics, aren't you, which is my strong subject. But that if the rest of the world is in danger, we're in danger as well. But that last slide that I showed you, I mean, these variants are being imported. So it would be to our advantage as well to have the rest of the world vaccinated. Now,
Right.
we are a member of this so-called COVAX scheme, which is supposed to send vaccines to developing countries. But I think that that so far anyway, that scheme seems to have operated very inefficiently, and it's the rich countries are hogging all the vaccine supplies. Yes.
Okay.
Yes.
Thank you.
Thank you, Sally. David Nunn.
Thank you, David, and thank you, Greg. Very interesting update there. I don't think you're going to want to answer this one, Greg, because it's sort of possibly touching into the politics, and it might not sound particularly humanist of me either. But then again, I've always maintained I'm an atheist and not a humanist. But that's neither here nor there. But you know, since this Indian variant was discovered, I just do not understand for the life of me. Firstly, how people were visiting India in the first place when I hadn't seen my mum for 10 months, and secondly, why on earth were they allowed to come back? Why were their flights coming back? Why did they? I just do not understand why this is being allowed. If I'm not mistaken, the Spanish flu back in the early, about 100 years ago, there wasn't international flights like there are today. So I presume that had an immediate break on the spreading of the virus. But it seems utter madness that people are flying to India in the first place and then flying back, and clearly have caused those blips you saw on your map in high population areas of Indians. It just beggars belief. Anyway, that was more. I just wondered, do you have a view as to why didn't they just say, right, stop flights coming in, stop? And would that have had an impact? Would it have stopped it coming in?
I'm not a politician. I'm a virologist, but yeah, I quite agree with you. I mean, it's difficult to see the logic because countries that have been successful in controlling coronavirus, you could think, for example, of New Zealand, have had much more stringent immigration controls than we have. And you can see that Indian variant happening. You can see the way it's, where the outbreaks have originated at multiple points throughout the country. You're quite right. The obvious way to control it is to stop people visiting India. And I don't know why that wasn't done. So,
well, I guess the visiting in the India is not a problem. It's them coming back.
Yes. Well, yes. Yes.
And I don't mean that in any sort of xenophobic way. But I mean, frankly, as I said, I don't understand. I thought we were in lockdown. I didn't think people could just fly to India if they wanted to. But as it turns out, they have been. And it's quite astonishing, really. Anyway, thank you, David. Thank you, Greg. Thanks again.
Okay. Thanks, David. Let me just pick up on something that Phil James put in the chat. Greg, I think Phil was asking about the vitamin D3 and whether this is an effective remedy or how it works.
Yeah, I don't know. This is something you see on social media. You see lots of things on social media, I suppose, but I'm not aware of. Phil might have more information about this than I do, but I'm not aware of any clinical trials that have been done on vitamin D at all.
Okay.
And I tend to, being a skeptic, I tend to only believe things that have got a scientific basis in terms of clinical trials. But
well,
I'm aware this has been touted. Yes, Phil. Sorry.
Yeah, no, it came from the Zoe. Gosh, what's it called? The Zoe app, the one that King's College Zoe app. Yeah, and there was some discussion on there. Apparently, they've done double-blind, randomized controlled trials in Spain.
I wasn't aware of that.
Yeah, and in France and Spain, they were giving high doses of calcifediol, which is, what D3 is turned into by the liver. And they were giving, like 1,000 units of calcividiol as a treatment for people with COVID, and they were seeing allegedly they were seeing some effect. But the question was whether there is actually a preventative issue, and of course the HM government is giving low dose vitamin D3 to clinically vulnerable people, so you can get that for free from the NHS. So there doesn't seem to be any. I mean, these clinical trials, they were fairly low. They weren't big, samples.
Yeah.
And
That's interesting. Yeah.
But there seemed to be some quite a lot of actual, reasonable evidence, but just not enough evidence.
Oh, I wasn't aware of that. That sounds interesting. I know from my own experience that older people like me do often have vitamin D deficiency. I mean, I take a vitamin D capsule every day, not against coronavirus, but just to guard against the fact that I'm not going out in the sun and to keep up my vitamin D level, because lots of older people in particular don't have sufficient vitamin D.
And apparently also neither do quite a few ethnic minorities for some reason. And that would potentially be, some sort of correlation that might help explain to some degree why they seem to suffer more than white people.
I'll just say that I'll just pick up what Sophie's put in the chat. Tim Spector of the Zoe app has said that vitamin D is not effective. He's changed his mind as he originally advised taking it. So he appears to have changed his mind on that. But
well, he hasn't, because there was a discussion between him and I forget the name of the other guy, Campbell, I think it was, and they had quite a debate about it, because Campbell was very much a proponent of using vitamin D3, and he was the one who came up with all the clinical studies. Tim Spector is really still sitting on the fence.
Okay. Well,
okay.
That's interesting. Thank you very much, Phil. I didn't know that.
Well, for my sixth benefit.
Thanks, Phil. And don't forget, Aaron has put a plug in the chat that you can get free vitamin D by coming on our walk on Sunday. So let's go to Chris Smith. Chris, if you'd like to unmute yourself.
It's been very interesting, as I expected it would be. My question's quite a quick one. We have ordered or over-ordered vaccine doses, but do we actually have them in store? Because unless we already have them in store, the question of whether we should be giving them away is rather moot.
Yes, it's more complicated even than that, because the vaccines that we've ordered that I had on that table, some of them haven't been approved yet in this country. So they're not vaccines that are available at the moment. They're vaccines which are still under development and presumably are in the latter stages of clinical trial. So, but we still have, even the 3 approved vaccines, we have more on order than we're able to use. And I gather there is a suggestion now that some of that stockpile will be sent to other countries.
Yeah, but that's what I'm trying to really pin down, whether it's something on order, excuse me, or whether it is an existing stockpile. Unsurprisingly, this is because of my contacts in Uganda who have been awaiting vaccines, and I've tried to inform them and they inform me.
No, I don't think it's a stockpile. I think these are vaccines that are on order.
Yeah.
And they haven't been produced yet. Yes.
That makes sense to me, and just thinking of the sheer production line that has to come into play to get those numbers. No, yeah. That's it. I'm happy with that. Thank you.
Thanks, Chris. And Sophie also made a point in the chat that possibly in places like India, they don't necessarily have the infrastructure to give the vaccines, even if the vaccines turned up. So again, that's another complicating factor.
Oh, can I just say there, David, that India is the biggest vaccine producer in the world. It actually produces more vaccines than any other country.
Yeah.
And it was supposed to export them when the incidence of coronavirus was low, but now they're using their whole production capacity, which is causing complications in other countries.
Yeah, there's no point them exporting their vaccines to us and then we send them back again.
Yeah.
Okay, let's go to Everard. Everard, if you'd like to unmute yourself.
Can you hear me? Yeah.
Yeah.
I kind of keep up with what I think of as the common man in the street every so often. And this friend of mine just the other day said about, and this applies to this the Indian thing, and he said this lockdown, it's gone on too long. He said. Now, what I think he means is the novelty is worn off. He's fed up with it, and these people are just simply ignoring all the rules of what do you call it, the bubble and things, the number of people you can interact with. They simply ignore it because they got fed up with it. And if he's doing it, probably 99% of people out there are doing it. Unless they're forced to do something, they've got fed up with it. And I think, it's all right for sort of eggheads like us who tend to think about things and act accordingly. Most people don't, I think. And I think that's why people are flying out to India, because nobody's stopping them. Despite the fact that it's obvious that that's going to cause an outbreak. That's all.
Yeah, I think you have a good point there. There were no I don't know why that was, but David Nunn was quite right. There were no regulations in force, even though it was apparent that there was this huge increase in cases in India. It wasn't on the red list of countries for some reason, so people could go there and come back with impunity, no problem.
Okay, thank you, Everard. Greg, Dan put something in the chat a little bit earlier, just asking for some kind of comparison between the immunity response from the vaccine as opposed to catching the virus and then getting immunity. Yeah. Is there a kind of comparison of those two things?
Yes, I should have given a bit more detail there, actually. At one stage, before the vaccines were used, you could tell whether somebody obviously had the virus infection by detecting immunity, particularly antibodies. In other words, to use serology, you could tell quite simply whether they'd had an infection or not. So we could tell if you measured antibodies in a group of people, you could tell how many of them have actually had an infection. Now, that's no longer possible easily. It is possible, actually, because, as I showed you, the vast majority of vaccines, apart from the Chinese one at the moment, consist of just one protein. So antibodies are only being made against the spike protein. Now, a natural virus infection produces immunity to lots of different virus proteins, not just the spike protein. So if you carry out a complicated analysis of not just neutralizing antibodies, you can tell whether somebody's had a natural infection or their immunity is due to the vaccine. But it's quite laborious.
And would natural immunity, is that likely to be more effective or less effective than a vaccine?
I don't think there's much data on that, actually.
Right.
The small amount of, no, I haven't actually seen the data. It has been claimed by vaccine manufacturers that the immunity particularly to the RNA vaccines, is better. But I haven't seen any data on that, and I don't think Tim has either.
Right, right, okay. I've got a question going back some months, actually, in terms of the politics in a way, but you'll remember, Greg, that probably back in September, there was this thing called the Great Barrington Declaration, and people like Professor Sunetra Gupta were involved in that. I mean, have those people been completely discredited, or what was that all about? I mean, I guess they were epidemiologists rather than virologists, but have they been totally discredited, or was there something in that?
I don't know whether you heard the news today, Dominic Cummings, but that has resurfaced because it seems to have been Boris Johnson's idea before he decided he wanted lockdowns. So did you not see that on the news? You should get heard. The original idea apparently that he had was that you should cocoon people like me, people in risk groups, let everybody else catch the virus, and herd immunity would develop. And I think that was really the basis of the Great Barrington Declaration, wasn't it?
Yeah.
Unfortunately, you can't cocoon everybody, and that's not the way it would work. And in fact, it probably would have caused even more deaths than actually occurred. We can't say for sure, but I think the lockdown, compared to other countries that were successful in containing the virus, our lockdown was very late. So I don't know. My own personal opinion is that herd immunity like that wouldn't have worked. But anyway.
Okay. Let's go back to David Nunn then. David.
Thanks ever so much, David. It's actually in relation to what has just been said. I remember quite distinctly on one of those wonderful BBC, news coverage with the Prime Minister in the middle and his two scientists flanking him right and left. And Sir Patrick Vallance turned around and said quite early on, herd immunity. It was. I remember it quite specifically. So I actually do believe that this was a scientific and government ambition. I mean, Sir Patrick Vallance has actually rode back a bit, saying, "Oh, I didn't say let's go for it." He said it's an option. In fact, actually, if I'm not mistaken, we are actually going to be getting herd immunity by vaccine and people having immunity from getting it. So effectively, we will get herd immunity. It's just that they were trying to get it very, very early on with the consequent deaths that would have resulted.
Yeah, but herd immunity from a vaccine is quite a different proposition because it doesn't kill anybody. Herd immunity from the way he proposed it would have killed thousands of people.
Yeah. Do you remember Sir Patrick Vallance saying that?
Yes. There's actually been a clip broadcast on the BBC showing him actually saying it. Yeah.
Oh, I haven't seen that recently. I do remember it from when he said it. I thought then, crikey, that's dangerous.
Yes, he did say it. Yes. He seems to have changed his mind later. I think countries that did go for lockdown very early on were actually the most successful at controlling the virus. So the proof of the pudding is in the eating, really, isn't it?
Thanks, Greg.
Greg, there's another question from Sophie in the chat. Does Gregory think that the AstraZeneca, Pfizer, etc., will be able to anticipate what the impact of the variants will be on vaccine modification and booster jabs? Will they be able to catch up with the virus evolving at high speed and the need to produce and test a vaccine with potentially quite different spike proteins? Have we made our vaccines too specific for the sake of speed and suffer because of lack of general impact?
Good question, Sophie. I think nobody can answer that. No, nobody at all. You can only really draw analogies to what's happened with other viruses, and the real analogy here is influenza virus, isn't it? Influenza is also an RNA virus, although it's a negative strand up virus. It's not a positive strand like coronavirus, but it produces variants at quite high frequency, so that we have to have a different vaccine every year to get round the escape of the virus from pre-existing immunity. Now, in terms of the vaccines, that is one very useful aspect of these new genetically engineered vaccines, because they can be engineered relatively simply and relatively quickly to cope with new variants. But what it would mean I have to say that there is no concrete evidence yet that the coronavirus has produced important escape variants. It hasn't. There are a few variants that show some degree of escape, but they don't show total escape, like the Indian variant, for example. The vaccine is still effective against it, albeit less so. But you could envisage a situation developing where the selection pressure on the virus is going to produce such escape variants. And if that happens, it would be possible for all the genetically engineered vaccines that are available at the moment to tweak them to include the variant sequence quite quickly. But it would also mean that everybody's got to be revaccinated, which is quite a logistical problem. It's done for flu virus, so it could be done for coronavirus. But Sophie's hit the nail on the head. That is, at the moment, is the $64,000 question. We don't know whether it's going to happen or not. We might be lucky.
Is that satisfied you, Sophie, as an answer? Yeah. Okay. Good. Greg, I've got quite a simple question for you, but I'd like to hear the answer from a scientist. When we go and get vaccinated, are we primarily doing it to protect ourselves or to protect other people? I mean, obviously we're protecting ourselves, but are we also protecting other people in terms of the vaccine making us not spreading it? I mean, is that part of what a vaccine does?
Absolutely.
Right.
As you go down the age group, I think the vaccine's got down to 30-year-olds now, hasn't it? If you take people of that age, under 30, the effect of the virus on them is minimal. They're not going to. They stand a very much reduced chance of being severely affected or dying from the virus, although they do get a standard chance, but it's very much lower than for older people. So really, the risk to them is quite minimal, but they're protecting other people who might be susceptible and who might die from the virus. So
okay, so how does that work? How does the vaccine, it kind of kills off the virus in your body, does it? Is that what happens?
No, it's more complicated than that. It's not the virus, it's not the vaccine that kills it off, it's the immune response to the vaccine.
Right.
So
okay,
it's a combination of the different arms of the immune system being stimulated artificially. What's happening is your immune system is reacting as if you've got a virus infection, but it's been deceived. There's no virus infection, there's a vaccine. So it's not the vaccine that's doing it, it's your immune system.
Right.
And of course, different people react in different ways in terms of their immune system. David, I had a correspondence with you on this. Your aunt or somebody was one of these very few people who developed no antibodies to coronavirus. Is that right?
That is right, yeah. And she's causing it. She happens to live in the constituency of Theresa May, and she's causing a right stink about it because she thinks this is like a gap they've missed.
Yes, but as I said in my talk, antibodies are not the only arm of the immune system that's important here. In fact, it might be that cell-mediated immunity is more important, and we don't know whether she had any cell-mediated immunity, do we?
No. Frankly, I think she's just stirring the pot, really.
So, okay, so Greg, we should, are you saying we should go through vaccinating the entire population, including children? Is that what we should be doing?
Yeah, it's not really a scientific question here, it's an ethical question, because when you get down to children, teenagers and people like that, they really have an extremely low chance of being affected by the virus. Right. So really you're doing it to protect the population in general, not to protect them.
Right. And, so it becomes an ethical question.
This business of blood clots is a similar kind of balance of risks, because it is 1 in 2 million people will die from a blood clot after being injected with the Astrozeneca vaccine. Very low frequency compared to, say, what happens with measles vaccine, for example. But there's still, for older people, there's a very considerable risk benefit. But when it gets down to younger people, the chances of them dying of a blood clot are approaching the chances of them dying from the virus. So you have the risk balance is altered.
Oh, right.
Really an ethical question then.
Okay. And what about viruses causing an autoimmune reaction that is dangerous?
Yeah, this is an even rarer effect of viruses. It's something that's known to occur with measles virus, but it's an extremely rare side effect and got to be balanced against the risk of that people have from the virus itself.
Yeah.
So it's really a risk assessment.
Yeah. Okay. Yes, it does. It does appear that we've had 2 famous people in the audience. Carrie Simmons was here earlier, and she seems to have mutated into Dominic Cummings. So I'm probably not the real Dominic Cummings, but anyway, it's good to see you, Dominic. Well, Greg, this has been very informative. It's been stretching, scientifically for those of us who are not scientists, but fascinating nonetheless. Unless there are any other questions that I've missed. Oh, Lizzie's put something in. Lizzie is asking, is there any confirmed data on women suffering from the side effects of AstraZeneca compared to men? And why do some people suffer no side effects or reaction to the vaccine and others suffer expected or severe effects? I don't know if you can say something briefly about that, Greg.
I don't know of any data on gender effects on side effects of the vaccine. I think the side effects are so low in frequency that it will be difficult to measure anyway. There is a gender effect of the virus in terms of deaths, but I don't know any of, I'm not aware of any for the vaccine.
Okay.
Okay.
Thank you. Sorry about that.
That's all right. That's okay, Lizzie. And Phil Warburton, you're saying you posted a question earlier. Phil, do you want to just ask your question rather than me try and find it?
Yeah, okay. On the ACE2 receptors, that's the receptors that the virus attacks. People take certain drugs for high blood pressure which block those receptors.
Yeah.
Specifically to stop the enzyme. I forgot what it's called now. So there's something enzyme. Would that drug stop the virus, or is it too tiny for it to have any effect?
Yeah, good question. I take that myself. ACE inhibitors, aren't they?
Yeah, ACE2. You can get one that inhibits the enzyme, and you can get one that blocks the receptor.
Yeah.
In fact, I'm taking that.
I have to say that there's been a lot of effects. I mean, there's no doubt about it that COVID-19 certainly has drastic effects on the circulatory system, in particular causes blood clots and things like that. Whether that's linked to this receptor, I just don't know. I've not seen any data on it. There's been some speculation, all right, but I've not seen any concrete data. So I have to
I was
say,
just wondering actually if it would actually physically stop the virus entering the receptor.
yeah, one would think so. One would think so, but surely somebody must have thought of that. But I've not seen
That's
any.
what I would have thought.
Well, Ivermectin is supposed to block the ACE2 receptor. That's what it does in cell culture, but it doesn't seem to work in an actual patient for some reason.
One thing I can say is I haven't had a flu injection since 2008, and I haven't caught flu, and I've been taking a blocking thing of that receptor since before that time. So I just wondered.
Well, that's not the flu receptor. The flu receptor is completely different.
Is it? Oh, okay.
It's not even a protein; it's a carbohydrate. But
I thought someone was
yes, that wouldn't be a mechanism.
No,
not for flu.
No.
I just don't know. One thing you have to say about this virus, we've only known about it since the year last November, so a lot of things are unknown, and I think physiology of the disease is one of the complete unknowns. Why people get the symptoms they do, I think there's no mechanistic explanation at the moment.
Too many variables, I guess.
Yeah.
Okay, thanks very much, Phil.
Okay, yeah, thank you.
One final question for you, Greg, and then we'll wrap up. Are you optimistic or pessimistic about our prospects in this country for getting back to normal in July and August?
Oh, God. You're asking, I didn't bring my crystal ball with me.
You can say pass or abstain if you'd rather. But
well, one would hope that the vaccine program would have, would have that benefit. One would hope.
Yeah. Yes. Okay. Well, that gives us a little bit of hope then. I'm sure everyone really appreciates the time you've spent on this, Greg. And if people would like to show your appreciation in some way, either physically or electronically, that would be great. But thank you very much, Greg. It's been most enlightening and stimulating. So thank you all very much, everyone, for coming along this evening. Don't forget our walk on Sunday, and our bulletin will be out next week with our upcoming events. And great to see everyone, and keep in touch.